Good morning, everyone. I'm on Kyung-Ah Cho at Jeonnam National University as a chair of this session. This session is composed with immune aging and brain aging. And our time is from start at 9 a.m. and we have to finish in 12 at p.m. because at that time some closed remarks will be held. So please keep the time. I will ring a bell and every speakers at 18 minutes. In case of Professor Pradran's case, I will ring a bell in 28 minutes, okay? So if I ring a ring, please summarize, close your speech. So, and if I speak my presentation, please let me know if how much time I use. Okay, so now I will introduce the first speakers, Professor Bali Pradran at Stanford University. Yesterday, he is very well introduced as a plenary lecturer. So I will skip more detailed information about so please.
They have this nice podium here. Thank you very much, Professor Kyung Cho. And good morning, everyone. It's a great pleasure for me to be here at this very interesting talk, a very interesting session. Yesterday, I spoke about our work on systems vaccinology. And today, I'm going to talk about something a little bit different, which I didn't talk about yesterday. And this is a new concept which we proposed. It's called integrated organ immunity. And let me explain what I mean by this. Now, in the field of vaccines, when you assess how vaccines are doing in the clinic, what one measures is the antibody response to vaccines. And you can see on that table, there are many, many vaccines that have been approved for human use. They all induce antibody responses, and this is what's measured to assess whether the vaccine is immunogenic, and regulatory agencies use antibody measurements as what you might call a correlate of protection. But as we all know in immunology, vaccines induce many different kinds of immune responses. So in this diagram, you can see that vaccination initially induces this early innate response, which is highly conserved. It's an ancient system of defense, billions of years old, highly conserved between frogs, fruit flies, slime molds, and politicians. And this is really quite ancient, and it's very non-specific, and it's rapid, and it acts within minutes to days of vaccination. Now, this is then followed by the adaptive immune system, which you know very well. This is the immune system that's made up of antibodies produced by B cells and T cells, which are the cells that kill infected cells. And this is much more recent evolutionarily, and much more recent than the innate immune system. Now, the adaptive immune system has this feature that we call immune memory. So this is the ability of your immune system to remember an encounter with a virus or a bacteria for years, sometimes for a whole lifetime, so that the next time you see the virus or bacteria, you have a much, much more rapid response, much bigger response. And so we wanted to understand the immune response in a holistic way, not just measuring antibody, not just measuring T cells, but measuring all the different aspects of the immune system and how it protects. And as I mentioned yesterday, and I'll just briefly mention it now, just to put this in context, our very first experiment on this was done in humans with the yellow fever vaccine. You know, the yellow fever vaccine is a very classic old vaccine that's about 70, 80 years old. And it's a highly efficacious vaccine. It gives you lifelong protection, amazing antibody responses, highly durable responses that can last for a whole lifetime, and incredibly high T cell responses. So we did this experiment in humans where we gave the yellow fever vaccine and followed the immune response for about 3 months. We used RNA sequencing to profile the change in the gene expression in blood in response to yellow fever vaccine, because this is inducing a strong innate response, and you can pick this up by looking at the gene expression changes. We also measured the T cell response and the antibody response, and you could see computationally, using bioinformatics, we could come up with a gene signature that was induced by this vaccine and highly correlated with the T cell response and the antibody response. And then using AI machine learning, we could actually use this gene signature to come up with the predictive signature that could predict the T cell response and the antibody response. So this paper that was published in 2008 November is the very first paper showing the application of the tools of genomics and proteomics or multiomics or systems biology to predicting vaccine responses in humans. And since then, we and a number of other groups have extended this approach to looking at many other vaccines. This is a meeting on aging, and as I mentioned yesterday, although we are not experts in aging, and I'm learning so much from you about aging and the immune system, we are very interested in understanding how vaccines are responding or not responding in the elderly population. And you know very well that in the elderly population, we get two different types of pneumococcal vaccines. Pneumovax, which is a carbohydrate vaccine, and Prevnar 13, which is a conjugate vaccine against pneumococcal disease. So we have done a study in humans where we look at young people between the age of 20 and 45, not so young people between 60 and 69, and not so young people between 70 and 89, okay? And we give the Prevnar or pneumococcal vaccine, and we collect blood samples at all these time points, and we follow them for about 3 years. And I just want to show you one data slide, and that is what is the immune system of a young person versus a not so young person look like. If I take a drop of blood from young people versus elderly, how will this immune system look like when you look gene expression profiling? And this is what you can see here. This is before you give the vaccine. This is what's called a heat map. And in this heat map here, you can see all the gene modules that are expressed. You know, if you look at it, this is very remarkable heterogeneity. These people seem to have a lot of red here. These people not so much red here. And it turns out that these people are mostly the young people, and these are people as you get older. I just want to make one point. That is, as you age, your immune system at baseline, that is before you give the vaccine, is becoming quite different, and you can use these tools to actually describe what's happening. And if you now go a little bit more into detail and ask what kind of genes are changing as you get older, you can see these are gene enrichment pathways that progress with age. So you see here, the yellow lines enriched in activated dendritic cells, chemokines and inflammatory molecules, inflammasome receptors and signaling. Those are all enriched in older people. So as you get older, your immune system is becoming more like an inflammatory signature. And you know that, that's nothing new. But in the next few slides, I'd like to tell you why we think and how we think this is happening, okay? So let me turn to the focus of this, and that is this question of integrated organ immunity. Now, I told you that in immunology, the concept of immune memory is based on memory T cells and memory B cells. But what's happened in the last 10 years is that there's a new idea that's been proposed, that this ancient system of defense called the innate immunity, that's made up of monocytes, dendritic cells, and so on, also can have a kind of memory. And my good friend and colleague Mihai Netia proposed this term, trained immunity. That is, the innate system, you can train it to remember. A little bit like memory T cells, memory B cells. So, and the basis of this training is thought to be epigenetics. Now, you all know about epigenetics, but this is a nice analogy. Imagine the genome or the chromosomes is like a book here. Now, each chapter in the book, the book is the whole genome. Each chapter in the book is a chromosome. Each page in the book is one gene in that chromosome. Now, if you want to read Act 1, Scene 1 of that book, that is a certain gene, you open to that page and you can read it. Another word for reading is transcription, transcribe, okay? But that is how genes are expressed. Epigenetics is all about having a bookmark. So the book is closed, it's not open, but you have a bookmark at a very specific page, this page, Macbeth Act 1, Scene 1. And in this way, you can quickly access that page. You know, aha, this is where I need to read. So you can very quickly, you don't have to waste time saying, is it page 300? Is it page 500? So this is the basis of epigenetic imprinting. So several years ago, when we moved to Stanford, I was interested to ask whether there is innate memory in the context of vaccines. So we did a study in which humans got this bird flu H5N1 vaccine with ASO3 adjuvant or the Pfizer-BioNTech COVID vaccine. And then using single-cell ATAC-seq. So this is a technique where you can measure the chromatin accessibility. That is, you can read the bookmarks to see which genes have these bookmarks. We isolated blood cells from people before they got the vaccine, and then 1 month or 2 months after they got the vaccine. And we isolated monocytes, and we did this ATAC-seq, and we found that certain regions of the chromatin targeted by certain transcription factors, like IRF1, STAT1, STAT2, were wide open. There were lots of bookmarks in those regions, meaning that those genes were ready to be expressed, okay? Why was this important? Well, it turns out, as you know, these transcription factors, IRF1, STAT1, STAT2, are important in antiviral immunity, that is, controlling viral infection. And so the other thing that was fascinating was that in the same cell, same monocyte, there was another region of the chromosome, another chapter in that book, which was very closed, no bookmarks. What chapter was that? That was the chapter targeted by AP1 transcription factors. CFOS, CJUN, JUNB, you know very well, these are transcription factors that induce inflammatory cytokines, IL-6, IL-12, TNF. So inflammation module is closed epigenetically, very hard to express. Antiviral module, IRF, STAT1, open epigenetically, very easy. So the obvious question was, so does this mean that this bird flu vaccine has primed the innate system to be more effective at broad antiviral defense against all kinds of viruses, not only bird flu, but maybe dengue or Zika? These are viruses that infect monocytes. So this is what we did. We were able to isolate from the blood of a person before they get the vaccine, the bird flu vaccine, or 2 months after they get the bird flu vaccine, and infect these cells with dengue or Zika. And what we showed was that 2 months after they got the vaccine, these monocytes could control dengue or Zika much more effectively. In other words, the bird flu vaccine had imprinted epigenetically on monocytes the ability of those cells to control a very different virus, dengue or Zika, okay? So this is an example of this trained immunity, epigenetic imprinting in the context of vaccines. Now, what about the other loci I mentioned, this AP1 loci which controls inflammation? I said that this is now closed 2 months after vaccination. So does that mean that there's less inflammation in these cells? And what we did was to isolate monocytes before you give the vaccine, 1 month or 2 months after you give the vaccine, and stimulate them with toll-like receptor ligand in vitro. And we measured cytokines. Guess what? In every human, every subject, each line is one subject, you can see the monocytes produce much less of these cytokines, these bad inflammatory cytokines. So it's actually suppressing inflammation. but promoting antiviral immunity, okay? So this was a very striking finding, but that was only with one bird flu vaccine. So what we did was to now do a similar analysis, but with a whole bunch of other vaccines. Bird flu vaccine here given with ASO3 adjuvant, the H5N1 vaccine without adjuvant, the seasonal flu vaccine, Pfizer-BioNTech vaccine, yellow fever vaccine, and the Shingrix vaccine given with ASO1B. And in each of these cases, when we looked at the epigenetic changes, the most important cell in your blood that is responding epigenetically is monocytes. You can see CM is classical monocytes. With this vaccine, the number of differentially accessible regions are here, same thing here. Now with Pfizer vaccine, there's T cells are also responding as it is with yellow fever, but really it's the monocyte. And so we looked more deeply at the monocyte changes, and what we can see is that if you do a global analysis of all the epigenetic changes that are happening across all 6 vaccines, different types of vaccines, and you ask, is there some sort of a global architecture to the epigenome of how the human epigenome is organized in response to vaccination? There seems to be this beautiful internal logic. So this heat map shows many different regions of the chromosome. So this is AP1 region targeted by CFOS, C-Jun, and it's red in color because this is becoming more accessible. It's opening. But look what happens when AP1 becomes more accessible. What about this IRF stat, or the antiviral program? Look, it's blue in color. Why? Because it's becoming less accessible. So there is a reciprocal relationship between accessibility of this AP1 module, inflammation module, TNF-IL6, and the antiviral module. We hear the word reciprocal in the context of tariffs, but so too with the epigenome, it's a reciprocal regulation. But look what happens when this IRF STAT1 module is widely open here, this red box here. So that means now these are open. What else is open? Well, CEBP, that's another module, which is CCAT enhancer binding protein. That's open as well. So these are coordinately regulated. So this is remarkable that in the human genome, that regions of the chromosome that are located at really very different places can come together coordinately. And the other thing I wanted to ask is, where are these epigenetic changes coming from? You know, monocytes, you know this, what is the half-life of a blood monocyte? 1 day, 2 days. So these are short-lived cells. What did I say? How long-lasting are the epigenetic changes? 2 months. Long-lived epigenetic changes in short-lived cells. That seems like a paradox. How can it be? So we asked, where are these monocytes differentiating from? They differentiate from these common myeloid precursor cells or granulocyte monocyte precursor cells in the bone marrow. So what we did was an experiment in humans where we isolated blood from humans before they got the vaccine or 1 month after they got the vaccine. We isolated bone marrow precursor cells, and we fact sorted CMP, common myeloid precursors, or GMP, granulocyte monocyte precursors. And from blood, we fact sorted CD14 plus monocytes. Then we did 3 different techniques. Single-cell RNA sequencing to look at the genes that are expressed in each of these populations. Single-cell ATAC-seq to look at the epigenetic changes. And one other technique called mitochondrial DNA sequencing. Let me explain. This is a technique whereby you can trace the lineage of cells. All eukaryotic cells have mitochondria, and inside the mitochondria there is the mitochondrial genome, which is about 18,500 nucleotides. One region of the mitochondrial genome comprised of hotspots, which are highly mutatable. Highly mutatable hotspots. Based on the pattern of expression in these hotspots, you can draw lineage relationships between progenitor cells and daughter cells. So what we did was this, and this is the experiment, as I said, blood and bone marrow samples before you give the vaccine and 30 days after you give the vaccine. So first question is, what do you see in the blood monocytes? Never mind bone marrow, let's just focus on blood monocytes. So in this volcano plot, you can see there you are, these interferon regulatory factors are opening up, as we have seen before, relative to day 0, but this AP1 transcription factors are closing. So that's very nice. That confirms our previous analysis. Now let's look at the bone marrow. Let's look at the common myeloid progenitor cells, the granulocyte monocyte progenitor cells, and there you go. You see the same profile, the epigenetic profile, as we saw with the CD14 plus monocytes, arguing that there is this epigenetic imprinting happening in the bone marrow at the precursor or the stem cell level. And this could explain why there's long-lasting epigenetics in short-lasting or short-lived cells in the blood. Now, we are in the process of doing lineage tracing, but I do want to mention to you who are interested in aging why this is of potential significance in the context of aging. Some really elegant work by this group at the Jackson Labs have shown in mice, this is so far in mice, that this AP1 module comprised of C phosphine June is highly expressed in myeloid cells in aged mice compared to young mice. And they also did ATAC-seq, and it's highly expressed in monocytes and macrophages from old mice compared to young mice. So the epigenome is open. And we are seeing with vaccines the opposite. That is the less accessibility, suppression of the AP1 module, and enhanced accessibility via RF1 loci. So we're now setting up an experiment in humans where we will do exactly what we have done with vaccines, but from elderly humans, young humans, collect blood, collect bone marrow, and do these epigenetics and mitochondrial DNA tracing to see whether we can see this. Because if we do, then the implications are you could modulate these epigenetic changes at the stem cell level in humans with epigenetic adjuvants. So that's where we're headed, okay? So in the remaining few minutes, I have talk to you about trained immunity and epigenetics, and you would probably think that this is the mechanism of trained immunity, that is innate memory. In fact, in the field, the words trained immunity and epigenetics have become synonymous. So when somebody mentions the word trained immunity, the immediate reaction is, it's epigenetics. Or when somebody mentions epigenetics in the immune system, they say, it's trained immunity. But I want to now essentially tell you everything I've said before is not really accurate. So basically I want to say it's not just epigenetics. And I just want to show you a few brief slides from a recent study that we published, but this is ongoing work. And this study concerns a vaccine which you may know very well. It's the TB vaccine called BCG. BCG is one of the oldest vaccines. It's been given to billions of children worldwide. In the developing world, every baby that's born on the day of birth gets the BCG vaccine. Now, it was developed 100 years ago. Now, so what is very interesting is that in the field epidemiological studies, BCG has been shown not only to be effective against TB, but also against other kinds of infections. Nobody has really known why BCG has this heterologous effects. So Audrey Lee, a PhD student in my lab, decided to investigate this. What she did was to give mice BCG vaccine. She gave it intramuscular, sub-Q, or intravenously. And then 7 days later, 14 days, 21 days, 28 days, 42 days later, she infected these mice with SARS-CoV-2. This is the B.1.351 strain, which has high affinity for the mouse ACE2 receptor. So it causes infection in mice. And then she measured weight loss. And you can see here that 7 days, there's no difference, 14 days, no difference. But look, at 21 days later, mice that receive the BCG IV are completely protected against weight loss. 28 days later, they're protected. 42 days later, they're protected. And 3 months later, even, they are significantly protected. What was more impressive is when she looked at the virus, how much SARS-CoV-2 virus is there in the lung. And in blue, you can see our mice that did not get BCG, but that were infected with COVID. In red, our mice that did get the BCG, that were infected with COVID. 7 days later, no difference. 14 days later, maybe there's a slight difference. 21 days later, there's a 93-fold drop in the amount of SARS-CoV-2 virus in the lung. 28 days later, there's 435-fold drop. 42 days later, 92-fold drop. And even at 3 months, there's a very substantial drop. In other words, but if you look in the nose, not in the lung, but in the nose where the virus gets in, there's really no difference. So in other words, BCG is controlling virus replication in the lower respiratory tract, in the lung, but it is not preventing you from getting infected, okay? That's an important point. So Audrey said, how is this happening? How is BCG, a TB vaccine, able to control viral infections like COVID? So what she looked was serum cytokines in these mice, interferon gamma, TNF, RANTES. She looked at before you give the BCG, 6 hours after you give it, 1 day, 7 days, and so on, up to 7 weeks, or 6 weeks. And what she can see is that there's delayed induction of interferon gamma, TNF, RANTES, IL-22, but also she sees in many cases this early brisk induction of a little bit of IL-22, IL-7RA, MCP3, and so on. So there's this biphasic kinetics, the early innate response, which peaks within the first 24 hours, and you know that very well. Any vaccine induces an early innate response, but also this very second phase of this innate response that is more delayed and that lasts for several weeks. Why is that so? So Audrey then looked at T cells induced by BCG vaccine. These are BCG-specific T cells. So she removes the lungs of the mice 7 days later, 21 days later, 42 days later, stimulates the lung in vitro with BCG, and then looks at intracellular cytokine staining for CD4 T cells, CD8 T cells. And she could see that there's a very high magnitude, like almost like 5%, almost 6% of CD4 T cells make interferon gamma at 21 days, and even at 42 days, even at 3 months, there's this ongoing T cell response. So Audrey then said, okay, BCG is a complex bacteria. It has thousands of genes in its genome. It's probably activating many innate immune pathways. Which innate immune pathway is important in this cross-protective effects of BCG? So she immunized MyD88 knockout mice. You know, as immunologists, MyD88 is the main toll-like receptor adapter protein. ASC for inflammasome knockout and sting knockout. And she could show that the protective effects of BCG was completely abrogated in the MyD88 knockout mice. So no MyD88, BCG does not protect against COVID, okay? She could also show that the immune response induced by BCG, so that is the serum cytokines that I mentioned, like interferon gamma, TNF, and so on, was completely abrogated in the MyD88 knockout mice. And the T cell response induced by BCG was completely gone. So MyD88 is the dominant innate pathway through which BCG is mediating its cross-protective effects. Finally, Audrey said, is it possible that the CD4 T cell response induced by BCG is feeding back on the innate system to provide this protection? So Audrey immunized the mice with BCG, and before she infects with COVID, just a few days before, she injected anti-CD4, anti-CD8 antibodies to deplete the T cells. And what she could show was that if you deplete the CD4 T cells, you completely destroy the protective effects of BCG against COVID, okay? Now, if you get rid of CD8 T cells, no effect. But of course, if you get rid of both CD4 and CD8, you do abrogate the protective effects. Now, you might be wondering, wait, BCG is inducing T cells specific only to the BCG bacterium, and yet it's having an effect on protecting against COVID. Why? How is that? So, and again, it turns out that interferon gamma secreted by these CD4 T cells are important. If you neutralize interferon gamma, the protection is gone. TNF is not important. So the last 3 slides, I want to tell you the mechanism. So Audrey did a spatial transcriptomics. In other words, she got a whole lung from a mouse and looked at the expression of every single gene in every single cell in that mouse lung, okay? So this is one whole mouse lung of a normal resting mouse. It hasn't got the BCG vaccine. It hasn't been infected. And you can see green. Green is pancytokeratin. These are the epithelial cells, which are the targets of viral infection in the lung, whether it's flu or COVID. And in pink, you can see some CD4 T cells. Well, maybe not, because this is a resting mouse. There's not much CD4 T cells. Let's take a look at what happens 1 month after BCG vaccination. Let's take the lung out and do the same analysis, and you can see there's lots of CD4 T cells that have now migrated to the lung. These are T cells that we've seen against BCG. Now let's take a look at let's look at each of these T cell areas and see what genes are those CD4 T cells making. You can see the number one hit, the highest level of expression, is interferon gamma. I showed you in the previous few slides that BCG specific T cells by FACS are making interferon gamma. Now let's take a third group of mice where you immunize with BCG, and you, after a few days, after about 3 weeks, you get rid of the T cells in vivo with an anti-CD4 or anti-CD8 antibody. So no T cells. So there you are. If you do this, and you can see all the pink cells have gone because you have deliberately eliminated them. Now let's gate on these green cells, which are the epithelial cells, which are the cells which are infected by COVID and flu, and say, how has the gene expression pattern changed in those epithelial cells? Let's take a look. So in this heat map here, in blue are epithelial cell gene expression from resting unvaccinated mice, and you can see a lot of interferon-stimulated genes, but they're all blue, meaning they are not expressed. It's a resting mouse. On the far left, you can see what happens after BCG vaccination. The interferon-stimulated genes are highly expressed in these epithelial cells, up to about 2 months or 3 months. But in the middle is a mouse in which you immunize with BCG. After 3 or 4 weeks, you eliminated T cells. Guess what? The interferon-stimulated genes go away. So this tells you that BCG is inducing BCG-specific T cells in the lung, which makes interferon gamma that feeds back on these other cells, epithelial cells, to turn on this antiviral innate immunity program. And you can do your calculations and get look at your favorite gene like IFIT-M3, IRF1, IRF7 with very significant effects. So this is what I wanted to tell you about this concept of integrated organ immunity. I began my talk by saying that vaccines historically for the last several decades have been assessed based on the measure of an antibody response. Then I mentioned that our knowledge of the immune system is now such that we realize that what you need for protection is not just antibodies. You have T cells, you have innate. What I'd like to propose is that protection is not mediated by any single pathway, but by the synergistic interaction of all these different innate pathways. So I gave you one example with BCG, where you see this biphasic innate response, this early innate response at 1 day, and then this delayed innate response at a few weeks. This delayed innate response I mentioned was a consequence of the feedback of the Th1 cells making interferon gamma that then acted on other cells in the lung, including the epithelial cells, including type 2 alveolar pneumocysts, to turn on this antiviral program that could then maintain a degree of broad resistance against infection. Now, I've shown you this with COVID. What about other viruses? Well, with Ralph Barrick, we infected these mice with the BA.5 Omicron strain, protection against the original SARS virus. There's a mouse-adapted strain, protection. Another coronavirus, SHC014, protection. It even induces protection against allergic inflammation after 3 months against house dust mite. So BCG, immunized IV, after 3 months, you induce allergic response with house dust mite, and you look at Th2 cytokines, IL-5 is down, IL-13 is down, eosinophils is down. And would you believe it, it even induces protection in a model of DSS colitis. So you give this BCG, after 35 days you induce DSS colitis, and you look at weight loss, and you can see in the control mice they lose weight, but in the BCG immunized mice they are somewhat protected, and you can also measure colon length. So what it looks like is that this vaccine that's almost 100 years old, that's been given to billions of people worldwide, that is every baby in the developing world is given this on the day of birth, is having far more effects than what we thought it was having. So it is protecting against SARS-CoV-2, SARS, the SH014 coronavirus, influenza virus, house dust mite allergen. I didn't show you, it's also protecting against Staphylococcus aureus bacteria and against inflammatory bowel disease. This argues that there must be a common mechanism of immunity that is having these beneficial effects. Our goal is to understand the molecular basis of that common mechanism, because if you did that, you could design drugs that target this pathway that could have a broad and beneficial effects on protection, not only against infections, but also against allergens and possibly other inflammatory diseases. And to you, the question is, what implication does this have in the context of inflammaging? Could BCG also prevent inflammaging? If so, what could we learn about the mechanisms that control inflammaging? So this basically summarizes what I said, this integrated organ immunity. It's not just innate, not just B cells, but not T cells, but the integration of all of this acting together. So these are all the amazing people who did the work. Audrey Lee did all the BCG work. Florian Wimmers and Prabhu Arunachalam did the previous work on the epigenetics in humans. Thank you very much.
Thank you so much. I didn't know about the BCG can protect inflammaging and other disease, kind of colitis. That is so surprise data. Thank you so much. Now I will introduce about TRF5-dependent immunomodulation-based therapeutic strategies for aging and age-related disease. Immunoaging leads to two major problems, inflammaging and immunosenescence. Inflammaging contributes to the rise of chronic disease like cancer, Alzheimer's disease, metabolic disorders, and other our known disease. On the other hand, immunosenescence weakens the body's defense systems, causing higher infection risk, reduced vaccine responses, and even failure of immunotherapy for cancer. This is why we saw such a high mortality rate among the elderly peoples during the early days of the COVID-19 pandemics. Now, immunoaging is recognized as a key concept in both aging and vaccine research. If we can enhance and restore immune system function in the elderly using existing zero protective interventions may help overcome the low efficacies and high side effects of conventional treatment of drugs or vaccines. Actually, many well-known geroprotectors are now being tested in COVID-19 clinical trials. These studies are not just about treating the virus, they are actually aiming to improve immune functions in the elderly people. So through these clinical trials against infection is becoming a key concept to clinically validate longevity drugs. But they are not directly designed to target immunosenescence itself. So here is a question. If we can modulate directly modulate the immune systems, can we really change the aging process as anti-aging drugs? So kind of geroprotective interventions. My answer is yes. Last year, two important studies demonstrated lifespan extension by targeting immune aging. For developing of healthy longevity drugs, balancing of immune aging between inflammaging and immunosenescence is very important. One showed that inhibiting IL-11 can suppress inflammaging. And the other reveals that, that is our study, activating of TLR5 can overcome immunosenescence. Together, they highlight immune modulation as a promising strategy for healthy aging. In this study, the authors at Stanford used IL-11 knockout mice and IL-11 neutralizing antibodies to suppress IL-11 signals. They found that this led to a broad range of anti-aging effects. Blocking IL-11 reduced cell-specific factors and tissue fibrosis, which in turn suppressed chronic inflammation. As a result, metabolic function improved, fibrosis and senescence markers were suppressed, and most importantly, both health span and lifespan is dramatically enhanced. This is our study. We demonstrated that TLR5 activation improves immunaging and leads to similar rejuvenation effects like IL-11 inhibition. And IL-11 and TLR5 can regulate immunaging through distinct mechanism, kind of between inflammaging and immunosenescence. But interestingly, both lead to very similar outcome, including reversal of aging phenotypes and improved health spans, kind of metabolic enhancement and suppresses fibrosis. This suggests that directly targeting the immune system could be a highly effective strategy for controlling aging. Now I'd like to briefly introduce about TLR5. TLR5 is a well-known of innate immune receptor that recognize bacterial flagellin, and it's a key part of the innate immune systems, especially at mucosal surface like lung and gut. When activated TLR5, it doesn't just trigger inflammation. It plays a broader role supporting tissue repair, anti-inflammatory responses, and metabolic regulation. That's why its stimulation has shown anti-obesity, anti-cancer, and anti-fibrosis effect across different models. Like other known geroprotectors, TLR5 activation demonstrates broad systemic benefits across lifespan extension, immune function, metabolic enhancement, and cognitive health. Therefore, we propose TLR5 as a novel class of geroprotectors uniquely positioned to address immunosenescence. So far, all these results are based on activation of TLR5. So next question is, how about if TLR5 expression is suppressed? To explore this, we used TLR5 deficient mice. We tracked them as they aged. As they got older, they showed obesity, especially at 40 weeks. Compared to 10 weeks, there was a clear increase in liver fat accumulations, and genes related to lipid metabolism and fibrosis were also significantly unregulated in these mice. To address liver function, we isolated primary hepatocyte from these mice. Even without any external stimulus, we observed visible fat accumulation in the cells. Interestingly, genes related to fat uptake and gluconeogenesis were significantly increased, while lipogenesis genes were not notably unregulated. This suggests that liver dysfunction is driven more by excessive lipid absorption than by synthesis. Since the liver tissue displayed high expression of fibrosis-related genes, we isolated hepatic stellate cells for further analysis. These cells showed morphological abnormalities and a significant increase in fibrosis-related gene expression. So we also measured their mitochondrial metabolic activities, which is a marker of fibrotic activation. These results showed markedly elevated metabolic function, indicating strong activation of HSCs isolated from knockout mice. This TERA5 is deeply related in some metabolic disease conditions. So to evaluate the role of TERA5 stimulation in metabolic disease, we tested its effect in 3 different disease models. In a high-fat diet models, TERA5 ligand treatment significantly reduced obesity. In high-fat and high-sugar models, that is very well known, amylin diet induced obesity models. In this model, TERA5 stimulation also reduced both obesity and multiple metabolic markers. In the liver fibrosis model by induced methionine deficiency diet, it led to a notable decrease in fibrosis markers and significantly improved the liver enzyme levels, kind of AST, ALT. So we observed anti-aging effect and therapeutic efficacies in various disease models. Today I do not show the inflammatory bowel disease models, but various disease models, TERA5 stimulation showed very dramatically therapeutic effect. So to develop it as a drug, we have to optimize the flagellin. So now I will explain about that. Flagellin is composed of flagellum of bacteria. So bacteria, when bacteria move, flagellin monomer dissociated from flagellum and then recognized by TERA5 receptors. And these 2 extra domains of TERA5 combines 2 flagellin. So this is flagellin structures. Flagellin generally have 4 domains, 3 domains, and 2 domains. D0 and D1 domains is very essential to bind with TERA5. But D2 and D3 domains is exposed in outwards, so that is provides very high immunogenicity. So developing of drug or vaccine adjuvant of flagellin, we have to minimize of these immunogenic domains. But so I screened many strains of bacteria, flagellated bacteria, and then we choose this microbiome originated flagellin. This flagellin is just have D0 and D1 domains. Do not include D2, D3 domains. So we selected this flagellin and then minimized the immunogenic domains and T-cell, B-cell, removed the T-cell and B-cell epitopes. So we finally optimized the best safety and good ligand of flagellin. So now I will call this flagellin is MSP102. We compared MSP102 with the original flagellin, MSP101, used in earlier studies. MSP102 showed stronger effects across multiple outcomes, including lifespan extension, anti-obesity effects, and improvement in glucose tolerance and insulin sensitivity. Overall, MSP102 demonstrated superior therapeutic potential. So for clinical trials, we have tested the MSP102 in various infectious disease models as a vaccine adjuvant. In the COVID-19 infection models, it has been compared with other licensed adjuvant, alum, ASO3, or MF59. And this study is collaborated with Korean NIID. And it showed stronger antibody productions and improved survival, and especially a greater suppression of lung inflammation that lead to long COVID symptoms. So we also tested the vaccine adjuvant effect in an aging model challenged with influenza infection. The result was impressive. We observed a high survival rate, strong antibody production, and clear activation of mucosal immune systems in BARF and NARF. So to summarize up the result, TLR5 activation led to immune enhancement, health span extension, and therapeutic effects across multiple disease models. In contrast, when TLR5 expression was suppressed by knockdown, we observed metabolic dysfunction and degeneration of immune organs such as thymus. In the TLR5 dependent deficient models, liver fibrosis became much more severe when challenged with MC diet, along with a significant decline in liver function. We also found a marker of increase in senescence-related genes, especially those linked to the Sen-Mayo signatures, is suggested by Mayo Clinics, suggesting that loss of TLR5 may accelerate aging. These results have been accepted for online publication in Aging Cell. The main route of our materials administration is intranasal. Compared to injection, it showed stronger effects and was also much safer in preclinical models. It stays local, and it doesn't cross into the brain, so safety profiles is very strong. After intranasal delivery, TLR5 high-expressing dendritic cells can be activated in the mucosa and lymph node. And this TLR5 specific dendritic cells can trigger Treg that release IL-10 for anti-inflammatory functions, or Th17 that release IL-22 for tissue regeneration. Together, this makes MSP-102 a potent mucosal immune activator with potential for vaccine or anti-aging therapies. And our MSP-102 has been officially registered in the NIAID adjuvant database is compendium. It is also being tested through the ACES project, which is a large-scale adjuvant comparison and characterization project led by NIAID, and Professor Plandrans is also one of PI this project. So our material MSP-102 have been tested in cancer, infectious models, or allergies. So yesterday I heard from Professor Plandrans, our materials may be very good, activator of Th2 responsiveness and not so high inflammation. So I really have to hear about good news from you. So we will discuss about the more collaborating works. So previous Professor Plandran's presentation, his very systemic biology and immunology is very stronger. So he will test some epigenetic changes of immune cells before or after our material treatment with aging, so we can understand how TLR5 can regulate this kind of multiple effects through immune cells profiles. And another mechanism study, I am collaborating with Professor Dimitri Blavin in some at France. He discovered a unique population of P16 high-expressing immune cell subsets that increased with age. But this immune cells, unlike typical senescent cells, these are not harmful. Instead, they have protective and anti-inflammatory functions and promote disease tolerance in various disease animal models. So he published in bioRxiv and some revision process in immunity papers. Interestingly, he also found that TLR5 can induce same types of P16 immune cells. So based on this finding, he proposed a collaborative study. So, and our collaborating works is now in progress. So our materials have sent to his labs, and he will provide a very good animal models for monitoring of these P16 high-expressing immune cells model tested. So now this is my last slide. So through this multiple-phased research, we aim to go beyond traditional seroprotective xenomorphics by directly modulating the immune system itself. This approach focused on restoring immune homeostasis and balance, and we believe it could become a key strategy for extending healthy lifespan. So for this study, we are collaborating with domestic and abroad many collaborators. So I hope our materials can be a good seroprotective interventions by controlling our immune systems. Thank you so much. Next speaker is Dr. Ui-ho Kim from Pasteur Institute. He will give a message about impact of immune aging on respiratory infections.
Thank you very much for the warm introduction. I am grateful to the organizers for inviting me to this wonderful forum. And this occasion is especially very meaningful to me because Professor Baliplandran was my former boss during my postdoc. So yeah, it is very honorable and it makes me a little nervous. Yeah, just kidding. Like my respected mentor, I'm an immunologist studying viral infection and vaccination, and I knew that he's going to talk about like all vaccine stuff and immunology stuff. So I was trying to avoid that. So this time I'm going to give you a brief and general overview of how immune aging impacts respiratory viral infections. So why focus on aging in viral infections? So as we know, some important respiratory viral infections such as COVID-19 and influenza cause more severe symptoms and higher mortality rate in the elderly population. As shown here, the numbers of COVID-19 confirmed cases are not that different except the very young population. But the mortality and the case fatality rate increases dramatically with age starting from the age of 60s or 70s. In addition, the data from the Lancet journal also showed a strong trend of increasing mortality with age in influenza infection. So together, these data clearly demonstrate a close association between aging and protective immunity to respiratory viral infection. So now I'm going to talk about what happens to the immune system in the aged population. So cellular senescence is the most representative phenotype of aging. And it is a self-fate induced by cellular stress resulting in changes in gene expression, morphology, and metabolism, followed by growth arrest and resistance to cell death. And the senescence-associated secretory phenotypes called SASPs are mostly cytokines and factors triggering inflammation and also accelerating the others' senescence. And upon stresses during aging, such as genetic instability, telomere erosion, cellular senescence is induced. And the release of SASPs, it affects the neighboring cells and mediate the accumulation of senescent cells. And then finally, it results in the establishment of chronic inflammation and other functional defects. And this cartoon illustrates key features of immune aging. There are multiple layers to this process. With aging, changes occur in the hematopoiesis, that is immune cell development, especially like the myeloid-skewed hematopoiesis, as mentioned by Dr. Plandren. And also, along with the impaired function of the innate immune cells, including macrophages and dendritic cells. And also, the thymus attrition is very popular phenotype of immune aging, and homeostasis and all B cells and T cells were impaired during this immune aging. And these alterations contribute to several unfavorable outcomes in the body, including the chronic inflammation, also known as inflammaging, and impaired immune regulation, and reduced immune responses to infections and vaccination. And in the next few slides, I'll walk you through some important facts and recent findings highlighting the key features of immune aging. The first feature is myeloid biased immune cell development in the aged bone marrow. So the immune cell development starts from the hematopoietic stem cells in the bone marrow and then differentiate into two major progenitor cell types like lymphoid progenitors and myeloid progenitors. In the young hematopoietic stem cell development, it shows more preferred lymphoid progenitor development. But this pattern is reversed during aging and show the increased production of myeloid cell types, and then it promotes the inflammatory environment. And another well-known feature of immune aging is about T cells, and this figure shows the age-related thymic atrophy in mice, and especially in the adult and old ages, you can see very small thymus here. And as the thymus shrinks with age, the generation of new naive T cell is decreased, leading to an accumulation of effector and memory T cells with the increased clonality, then a diverse pool of naive T cells. And this shift contributes to the reduced responsiveness of aged T cells to pathogens and vaccines with senescence status and NK-like non-specific killing function and pro-inflammatory function and exhaustion. Aging affects not only the immune cell compartment, but also affect the supporting microenvironments, which undergo detrimental changes. The upper figure shows the impaired structure segregation between white and red pulp in aged mouse spleen, along with the increased extrafollicular B cell response, and also the accumulation of terminally differentiated cells like plasma cells in the lymph nodes. And the bottom figure illustrates the age-related loss of non-immune stromal cells in the secondary lymphoid organs, marked by the decrease of stromal cell marker, GP38 in red here. And as a result, the structural and numerical loss of stromal cells resulted in the reduced capability to support homeostasis and response of immune cells. Moreover, the recent studies have demonstrated that aging of immune cells significantly impact the non-immune cells and also contributes to the organismal aging. And in this study, a mouse model with the accelerated immune cell aging was used. They knocked out the DNA repairing enzyme in the immune cell compartment. And then the senescence of immune cells was confirmed by the markers like the cell cycle inhibitor P16, P21, and also SASPs, the MCP1 and IL-6. And interestingly, in these mice, P21 expression was elevated in nearly all organs, indicating that the aged immune cells promotes aging in non-immune tissues, followed by significantly decreased lifespan. To further validate the role of immune cells promotes aging, the systemic aging, the authors transferred all the immune cells to young mice and they tracked the senescence status. And in here, you can see in all the various organs, you can see the increase of senescence marker and also decrease of longevity of these mice. Conversely, when old mice received immune cells from the young donors, the levels of both senescence and the inflammation were reduced. Okay, then what happens in the aged lung? The aged lung lost elasticity and becomes stiffer, leading to the reduced pulmonary function. And in addition, the aged airway epithelial cells and alveolar macrophages contribute to the chronic inflammation in the lung. And during the respiratory viral infection, viral clearance is delayed due to the inefficient antiviral immunity, like the type 1 interferon response. And this can lead to persistent hyperinflammation, which may escalate to a cytokine storm in some cases. And the repair mechanisms for the damaged lung become less efficient. So now it is a good time to talk about some hope, the strategies to overcome immune aging. There are two major strategies to overcome the immune aging. So first one is targeting tissue environment, like the senescence environment, and then the second is to directly targeting the immune system to boost the immune response. And first, targeting the senescence cell or aging-related signaling pathways by senolytic drugs or xenomorphic drugs. And senolytic drugs can eliminate the senescent cells itself, and also the xenomorphic drugs can suppress the harmful effect from the senescent cells. And the plant flavonoids, like the phycetin and quercetins, are known to have the senolytic effect and also the tyrosine kinase inhibitor dasatinib is also known senolytic drug. And the monotherapy or combinatory therapy of these senolytic drugs are in the ongoing phase 1 or 2 trials, mostly for the inflammatory disease in the aged population. And second, directly boosting the immune system is the other strategy. And to this end, immunotherapy, like the checkpoint blockade or CAR-T therapy or vaccination, could be utilized for this purpose. And I'll show you some supporting evidences for these overcoming approaches. And this data demonstrate the senolytic effects of dasatinib and quercetin. In vitro, when they treated the combinatory treatment, it could selectively eliminate the senescent cell without significantly affecting the normal cells. And in vivo, this senolytic treatment in mice led to the increased lifespan and improved physical performance described by the treadmill endurance measures. And the bottom data show that the xenomorphic treatment, in this case, the mTOR complex 1 inhibitor, like the rapamycin. And here you can see the decrease of infection frequency when when people were treated with these xenomorphic drugs. And it turned out that upon the treatment of this rapamycin, like mTOR complex 1 inhibitor, they showed the increase of type 1 interferon response. And also, additionally, the treatment of mTOR complex 1 inhibitors has been shown to partially restore the flu vaccine-induced immune responses in humans. This is another approach to rejuvenate aged immunity. As mentioned earlier, the myeloid-biased homeostasis is one of the features of the immune aging. And this group identified the representative markers for the myeloid-skewed hematopoietic stem cell progenitor cells like this, and then they utilized the depleting antibodies to deplete these like the cells expressing these markers. So when they treat these depleting antibodies, they could selectively eliminate the myeloid biased progenitor cells. And in vivo, when they treated this drug, they could reduce the myeloid development and also increase the development of common lymphoid progenitor cell and also naive T cell population, along with the decrease of systemic inflammation measured by the expression of IL-1 alpha. Following infection with mouse-friend virus, the drug-treated aged mice showed enhanced antiviral CD8 T cell responses and with the reduced viral replication in the spleen. Lastly, I'd like to share an example of directly targeting the immune system to overcome the age-associated immune senescence, and that is vaccination. In 2015 immunity paper, Dr. Plundren's group clearly demonstrate a trend of diminished flu vaccine-specific antibody response with age in human. To overcome the impaired immune response, now the enhanced flu vaccines are available for all older adults, including the formulations with higher antigen dose or with adjuvants that boost immune responses. While many studies have explored this area, including Dr. Plundren's lab, I'm presenting data from my collaborative study with Professor Jo Kyung-ah, and actually she already showed some part of this data. Just conceptually, using the adjuvant, intranasal TLR5 agonist adjuvant with the flu antigen in the aged mice, we can see the increased vaccine response, like the serum antibody level is increased, and also secretory, the mucosal antibody level was enhanced, and along with the significant increase of neutralizing antibody and much better protection than without the vaccine without adjuvant. Okay, so take-home messages. Immune aging and immune senescence results from dysfunction in immune cells and their supporting microenvironments. And it also affects non-immune organs. And aged lung exhibits reduced pulmonary function. Upon infection, it mounts an impaired antiviral immune response along with excessive inflammation. And targeting senescent cells and boosting immunity are promising strategies to reverse aging-related defects and restore immune competence. Thank you very much for your attention.
Thank you so much, Dr. Yoo Kim. Okay, now we will start the next speakers. Professor Kyung-ho Choi at Seoul National University College of Medicine. He will introduce about enhanced CAR T-cell therapy using a novel gene modification.
Well, thank you for a nice introduction. So it's my great pleasure to talk about my data in this prestigious meeting. So please accept my apology for not having any data on aging, but it's on the CAR T cells against cancer. So please take this time as a kind of refreshing session away from the aging. So, so this is my COI disclosure. So CAR T cells, many of you may know that this is a kind of gene-modified cell therapy against cancer. So you isolate the peripheral blood T cells from patient and introduce antibody-based cell signaling structure called chimeric antigen receptor, and then reinfuse them to the cancer patient to treat cancer. So the CAR looks like this. So this antibody, the recognized tumor antigen, the single chain FB linked to signaling domain of T cell receptor. So when these CAR structure is enclosed to the lentivirus or retrovirus vector, and then introduce that as to the T cells, then if delivery efficiency is like 50%, then suddenly all of a sudden your peripheral blood T cells, 50% of your peripheral blood T cells will become CAR T cells that can kill the tumor cells. So this CAR T cell therapy has been proven effective in 2017 against acute lymphoblastic leukemia. So and then that led to FDA approval of two drugs, Kymriah and Yescarta, and now 6 CAR T drugs are commercially available for hematological malignancy in USA. But you can, as you can see, that most of this CAR T drugs are against the hematological malignancies, but the hematological malignancies consists of just 5 to 10% of the total malignancies, and all the solid tumors like lung cancer and colon cancer and things like that has not been very effective. The CAR T cells are not been very effective for those tumors. The reason may be these solid tumors have a mass and it has an immunosuppressive microenvironment, such as the regulatory T cells and MDSCs and suppressive cytokines such as IL-10 and TGF-beta. So even for hematological malignancies, compared to leukemia, the complete remission rate of lymphoma is a little bit lower, it's like 50%. But long-term remission is around 40%. That means 60% of CAR T-treated patients will have recurred tumor and eventually cannot be cured. Thus, functional efficacy of CAR T cells needs to be greatly improved for generalization for more broad indications, including solid tumors. So I'll introduce two CAR T enhancing platforms today. So one is ClipCAR and the other is Converter. I'll start from a clip. So when you look at the CAR structure very closely, so as I told you, the antibody part is extracellular, and it linked to the cytoplasmic signaling domains such as CD3 zeta, and linked with a transmembrane domain, which doesn't have any functionality, is just a physical linker. And then people are trying to enhance the CAR T cell activity by adding another cytokine stimulatory domains like CD20 and 4-1BB, which we call costimulatory molecule, and then adding more and more. And now there's a third generation, but now commercially available CAR T cells have CD28 or 4-1BB domain, which is Kymriah and Yescarta. But there's a little attempt to add any functionality to the linker domain, such as transmembrane domain in this study. So this work is originated from the basic study on the surface molecule called the CD99 by a collaborator in our department, Professor Eun-Young Choi. Actually, she's in the audience now. So she was working on a molecule called CD99 on T cells. And when T cells meet the tumor cells or antigen-presenting cells, this contact region forms a structure called immune synapse. So this immune synapse formation is very important for CAR T cell activation itself. And this structural immune synapse formation needs proper actin rearrangement and then microtubule interaction together. So when you look at this immune synapse structure from the bottom, and then so the actin actually encloses the tumor cells broadly, which is called the lamellipodia formation. So you can see the broadening of actin structures slowly, and so lamellipodia formation is very good. And then if you look at the microtubule, then the microtubule organizing center called MTOC is located centrally, and then it generates a lot of microtubules, and these microtubules interact with actin closely. But in the absence of CD99, actually you don't see that lamellipodia formation and not the central localization of MTOC, and then you don't see a very good microtubule formation, and the microtubule and actin doesn't interact well. So, and then if you look at the structural the T-cell receptor beta in an antigen molecule localization to the contact site. The CD9, in the absence of CD99, that contact has been lost, so this recruitment of crucial T-cell receptor-related molecules are not obtained. So, and then if you look at the domain studies, then so she made a cytoplasmic mutant domain, which doesn't have cytoplasmic CD99 domain, and transmembrane mutant domain, which doesn't have the transmembrane domain. And for the cytoplasmic mutant domain, actually you can see that this is CD99, and in CD99 co-localized with microtubule well, but does not co-localize with actin well. And for the transmembrane domain, it co-localizes with actin, but does not co-localize with microtubule distantly. So that means this transmembrane domain is important for microtubule interaction, and cytoplasmic domain is important for actin interaction. Especially we found that short amino acid stretch in the cytoplasmic domain, we call it just a membrane domain, is important for that actin interaction. And in the absence of CD99, tubulin and actin doesn't co-immunoprecipitate. That means that this CD99 mediate actin and microtubule interaction. So CD99 transmembrane juxtamembrane domain is critical for actin microtubule interaction and immune synapse formation. So using that information, probably we thought that replacing this functionless transmembrane domain with the CD99 transmembrane domain may enhance the immune synapse formation and may embrace the tumor cells more broadly and increase T cell activation. But when you look at the co-culture of this CAR T cells and tumor cells, actually this red one is CAR T cells and blue one is tumor cells. For one tumor cells, there are 2 or 3 CAR T cells are kind of enclosing those tumor cells. So this conjugation rate is greatly increased. And if you look at the activation of CAR T cells, it actually gets more activated by measuring by CD25 activation marker on the surface. So we call this as a clamping-based immune synapse potentiating CAR. In abbreviation, we call it the CLIP CAR. So if you look at the actual actin microtubule recruitment, and this for the CLIP CAR, we compared with the conventional CAR T cells, which is Kymriah. Compared to Kymriah, this CLIP CAR showed a better actin recruitment. So we call the actin polarization. And then MTLC should translocate to the contact site. And it's faster translocation of MTLC to the contact site, which MTLC translocation time is decreased. As a consequence, if you look at the in vivo data, then so the Kymriah-like CAR T cells actually kill the tumor cells quite well. This is a lymphoma model, but does not eliminate all the tumor cells, and eventually the old mice were dead. But with CLIP CAR confirmation, actually we eliminated all the tumor cells, and the old mice survived. If you reduce the cell number 1/5, then at this dose, the conventional CAR T cells doesn't show a great efficacy, but still our CAR T cells showed a 100% elimination of tumor cells. That means 5-fold enhanced efficacy. If you go to the solid tumor model, then the same thing happened. This is stomach cancer and pancreatic cancer. So with just 5 million cells, this CAR T is quite effective with just plain confirmation. But if you go to the 1/5 cell dose, 1 million cells, it gets less effective. But with CLIP CAR, it gets 100% effective. That means more than around 5-fold enhanced efficacy. So based on this data, so we kind of founded a company called Tickers. So Eun-young and I are co-founders of this company. So we we initiated a clinical trial for this CLIP CAR against CD19 positive lymphoma, and this phase 1 clinical trial is ongoing on 5 major hospitals in Korea. So summarize first part is the clip CAR enhances CAR T cell efficacy by immune synapse stabilizing using novel CAR backbone, and this is kind of general platform to enhancing CAR T cell activity. The first pipeline is against the lymphoma. The second pipeline is against the solid tumors. And then the converter CAR technology. So CTLA-4 is a very famous T-cell inhibiting molecule. This is target of first immune checkpoint inhibitor called ipilimumab, anti-CTLA-4 antibody. But anti-CTLA-4 antibody actually enhances the T-cell activity overall, so it enhances a lot of autoimmune T-cell activation, which raises autoimmune side effect. So if you have a certain measure that inhibits CTLA-4 just in T cells that you want, like tumor specific T cells, that would be good. So we try to develop the dominant negative CTLA-4. So here, CTLA-4 actually recognizes B7 on the antigen presenting cells. But it has also B7 recognizing receptor called the CD28 in the opposite. So CD28 engages B7, it delivers positive signal. We call that co-stimulation because they need a T-cell receptor together. But after T-cell receptor engagement and co-stimulation engagement, then CTLA-4 comes up to negate CD28 by hijacking B7 and delivers inhibitory signal again. So we thought probably we can make the dominant negative CTLA-4 by just deleting the intracellular domain of CTLA-4 inhibitory motif, which is called the CTLA-4 decoy that has transmembrane and as extracellular domain of CTLA-4. But if you overexpress this decoy receptor to the T cells, then you will compete with CD28 in the first place and inhibit the T cells in the first place. So we thought probably because these two molecules are cousins, so we take this intracellular domain of CD28 and put it into decoy receptor and to make the CTLA-4 CD28 chimera. And when you overexpress this CD28 chimera, then it will compete with CD28 in the first place, but will deliver CD28 surrogate signal to the T cells. And then when the CTLA-4 comes up, then it will now compete with CTLA-4 to negate those CTLA-4 inhibitory receptor function. So we call the CD28 chimera as an abbreviation is CD28. So in the adoptive T-cell therapy model, we showed that it is very enhances adoptive T-cell therapy efficacy in lymphoma and melanoma model. So we applied this information to the CAR T cells. If CAR T cells has a CAR molecule together, this CD28, it can be incorporated in one retroviral vector, so it does not have any cost-increasing effect. So if you have a CD19 CAR and CD28 together in the same retrovirus vector, and then it will express a dual receptor, so CAR and CD28. So we use the syngeneic murine CAR T cell model. So here's a BALB-C CAR T cells made, and then injected into BALB-C lymphoma-bearing mice. So actually, the efficacy is very nice. And so if you inject 5 million T cells to the mice, tumor-bearing mice, then the Kymriah showed quite a good effect, but doesn't eliminate all the tumor cells. But with CD28 CAR T cells, actually eliminate all the tumor cells. If you reduce the dose to 1/5, 1 million cells, the Kymriah-like cells doesn't show any efficacy, but this CD28 modified T cells showed a great efficacy. This is accompanied in the CAR T cell expansion in the peripheral blood. CD8 T cells compared to plain CAR T cells greatly increased the CD8 T cells and maintained a persistent number of CAR T cells later. And CD4 T cells, the kinase was a little bit delayed, but it showed a dramatic expansion of CAR T cells. So if you look at the characteristics of these T cells, they of course, they can have a highly proliferative characteristics, but it also shows highly effector functions, and including aerobic glycolytic pathways and CD28 downstream pathway genes are upregulated. If you look at the tumor infiltrating T cells, and the T cell filter is greatly improved. And then if you look at the T cell phenotype, then in exhaustion-wise, so it's a reduced proportion of progenitor exhaust T cells, but it increased the effector-like exhausted T cells. But this effector-like exhausted T cells are not actually exhausted because they produce more interferon gamma and TNF-alpha, and even the progenitor exhausted T cells showed more cytokines, which showed this is less exhausted, is polyfunctional. But the question always, was in mind is that actually who provides B7 in CAR T cell setting? Because in the conventional T cells, actually the B7 expressed on the antigen-presenting cells. So when tumor-specific T cells recognize tumor antigen on APCs, in presence of B7, then it will deliver T cell signal plus CT20 co-stimulation. And then this activated T cells will move to the tumor microenvironment and kill the tumor cells, even if tumor cells are B7 negative. But CAR T cells actually bypass APC, and they directly recognize tumor antigen on the T cell surface. So then where's B7? So the answer will be then the tumor cell itself should have a B7, or APCs, the tumor environment, will provide B7 to the T cells, and then tumor cells, the antigen to the T cells, which needs a 3-cell interaction, which is very inefficient. So we tried to look at that. So even in the absence of we eliminated B7 on the tumor cells. But still, it showed a great effect. It shows a great expansion of CAR T cells. Then this is surprising, but is that the host B7? This is APCs. So we use this B7 knockout mice. The B7 has 2 isoforms, which is B7.1, which is CD80, and B7.2, which is CD86. So you should have a double knockout mice. So we use this double knockout mice. Actually, the efficacy a little bit decreased, but still have a substantial efficacy. And then, but CAR T cell expansion is greatly reduced, almost eliminated. So we look at this part first, and is it then the host macrophages or DCs are actually presenting? So we depleted macrophages. Still, you can see great efficacy and a lot of CAR T cell expansion. We depleted dendritic cells using CD11 TTR mice. Still showed a great efficacy and a lot of CAR T cell expansion. Then what else? It's B cells. So B cells express the B7 also. So we made a B cell specific double knockout host. And now you have a quite efficacy, and then you eliminated all the CAR T cell expansion. That means the B cell is the main part of the inducing this CAR T cell expansion. Then what about this part? So in the absence of host B7, still they showed quite effect. So the remaining cells, the tumor cells doesn't have this B7, and then the host doesn't have B7. Then what else? This is just the CAR T cells. So and then we used the CD88 double knockout CAR T cells. Now you eliminate all the effect. So that means B7 on CAR T cells is very important. But B7 is not usually recognized as a T cell expressing molecule. So if you look at the CAR T cells, then B7 is expressed, surprisingly. And then the expression level goes down, actually, by CT2 overexpression. So then why expression level goes down? So it's not because of reduced transcription or reduced internalization, because we checked that there was not. The other possibility is because CTLA4 is known to take out B7 from other cells. It's called the transendocytosis. So we thought probably that is the case. Then so we separated CD80 expressed cells and CT28 expression, 2 different populations to get this transendocyte phenotype. And for this CD80, so we put the GFP in the cytoplasmic domain to see whether this CT20 cells actually take up these cells. So we call that potentially Ripper cells, and this is Ripta cells. And then we co-culture them together, and then we look at the Ripper cells. Actually, we could see a lot of GFP transfer to the CT2 positive cells. That means transendocytosis has actually happened. then why is it important? Then what is the phenotype of this reduced B7H reduced CAR T cells? We look at the ripped T cell phenotype. So it has a little bit of proliferation increase and quite a good efficiency of increasing cytokines. But it's not enough, because if you have a CD20 together, then it showed a great increase of cytokine production and better proliferation. Means, other than this B7 depleting effect, it has own signaling effect to the T cells. And then is it possible that autonomous B7 CD20 ligation is possible, and then it gives a signaling to the T cells, which generate another cell line, it's a reporter cell line, that reflects the CD20 responsive GFP expression. So we injected this reporter cell line, CAR T cells, to the CD86 double knockout mice with the CD86 double knockout tumor cells. The only source of this B7 molecule is CAR T cell itself. In this case, actually, you can see increase of GFP expression. That means this signal is delivered. And if you use the B7 knockout CAR T cells, that signal enhancement was abolished. That means this autonomous signaling actually happened. So both transendocytosis-mediated CD86 downregulation and CD22 signaling T cells potentiate CAR T cells. That means CD20 CAR T cells utilize B cells for cell expansion and T cells for functional enhancement, which were unexpected cell type for ligand providers rather than dendritic cells and tumor cells. So finally, the human cells showed the B7 downregulation and far greater enhanced the CAR T cell efficacy and cell proliferation. So to summarize the second part, the CD20 CAR T cells show dramatic increase in CAR T cell function and efficacy based on unexpected ligands on T cell and B cells. And for the senescence a little bit, because it is well known that senescent T cells lose surface expression of CD20. It is a well-known fact. So if in the elderly patients, you isolate T cells, then they will have a lot of CD20 kind of negative cells. But by inducing this CD20 to those tissues, artificially will give CD20 signaling better, so then it will overcome the senescent phenotype, probably. So CD20 CAR T cells may be our next generation CAR T cell therapy. So this work was done in my laboratory and an Eunyoung Choice laboratory, and the grad student Gary Nam did all the CD99 work, and our student Seoul Lee did all this complicated mouse experiment on CD28. And the human CAR T cell part done is Dr. Hyung-bae Park in my laboratory. He recently moved to Tickers Incorporation. And then the clinical trial was done in my biotech company, Tickers. Thank you for your attention.
Thank you so much. Okay, next speakers. Now we will move next speakers. Next speaker is Professor Tae-Wan Kim at DGIST. His topics of brain aging stem cell therapy for Parkinson's disease. And please keep the time. After 18 minutes,
Sure.
I will ring a bell.
Okay, thank you. Thank you, organizer, for inviting me today here. It's my great pleasure to be here. Today, I'm going to talk about stem cell therapies for Parkinson's disease. Just let me brief introduction of the Parkinson's disease. And Parkinson's disease is the second most common age-dependent neurodegenerative disease. They show some movement disorder, some rigor, tremor, and poor balance and workings. Pathological hallmark of the Parkinson's disease is this progressive degeneration of these pigmented dopamine neurons in the midbrains, and appearance of this Lewy body, which is accumulation of the alpha-synucleins. These two pathological hallmarks are the main research of the area in the Parkinson's disease, and trying to understand why dopamine neurons are degenerated and why alpha-synuclein is accumulated. And ultimately, the blocking them how to fix and this degeneration and how to prevent and alpha-synuclein aggregations. There are two treatment options are available. One is the medication, L-DOPA, taking a pill, dopamine precursor. And second one is the deep brain stimulation, and stimulation of the electronic signal in the brain. And those two treatment options can relieve motor symptoms but cannot prevent the disease progressions. Therefore, there's no currently available therapeutic options yet. The etiology of the Parkinson's disease is mostly sporadic. Therefore, there are urgent need for model systems to understand the Parkinson's disease and may need some cell source to replace the lost neurons. And we think that the human pluripotent stem cell provide the multiple application in Parkinson's disease research and therapies because that the stem cell can provide unlimited disease-relevant cell, that is dopamine neurons, and also this cell can directly applied for cell replacement therapies. So my research has been focusing on the establishment of the protocol to generate the midbrain dopamine neurons to use for the cell therapies and also Parkinson's disease modeling. And today I'm going to talk about how we can use stem cell-based dopamine neurons for using to cell replacement therapies. So first question is, why do we consider the stem cell-based cell therapies to Parkinson's disease? And GWAS study and whole genome sequencing from the Parkinson's disease patient identified many gene mutations in Parkinson's disease patient. And following up, the study identified the many cellular dysfunctions, such as some mitochondrial dysfunctions and lysosomal dysfunction, and alpha-synuclein productions, and so on. And those cellular dysfunctions are also found in sporadic case in the dopamine neurons in Parkinson's disease. So maybe it may be difficult to fix all those dysfunctions to cure the Parkinson's disease. Also, it may be difficult to how to fix it. The idea of the Parkinson's cell replacement therapy is why not simply replace the new neurons to the lost neurons. So just why not simply transplant the new neurons to the Parkinson's disease that is lost in the dopamine neurons. And you may concern about the stem cell-based cell therapies, but actually there are many the clinical trials of cell replacement therapies to the many diseases beyond the Parkinson's disease. So also the cell replacement therapies to Parkinson's disease has very long histories. More than 30 years ago, there was clinical trials of cell replacement therapies using human fetal tissues. And as you can see here, the transplantation of the human fetal tissue was survived in the patient brain. Also, the innervated and released dopamines. As you can see, the fat F-dopa image. And before the transplantation, there's the yellow color, that is a low expression of the dopamine. But 1 year post-transplantation, the color shows more the red color, that is released dopamines. So it may work. So transplanted the cells that survived and innervated and released the dopamines. But you can imagine that using the fetal tissue has the very many issues, such as some ethics, also non-standard tissue processing. Also, they used about the heterogeneous cell populations, also insufficient cell number. But the fact is, some patients had some motor rescuing the benefit more than 15 years. Also, this patient did not have the medications. But some patient, it means the cell therapy works in some patient. So maybe we need for another cell source that is the robust and reproducible, and off the shelf cell source is needed. So we believe that the human stem cell derived dopamine neurons can be the promising cell source because it's going to be scalable and ethically acceptable, and we can inject defined cell types. So therefore we established the protocol to generate midbrain dopamine neurons from human pluripotent stem cell in clinically relevant conditions, and which we used defined medium. Also, we optimized Wnt activation for derivation of the dopamine neurons based on the mouse midbrain development, the high Wnt expression where the dopamine neurons spawn. And we call this protocol is Wnt boost. And resulting Wnt boost cell show the high expression of the midbrain markers and minimized off-target markers. And minimizing off-target marker is critically important for the clinical translations because due to the safety. And those cells are found in another cell graft, another group cell graft, but we did not see those the contaminating cell in the graft. Also, and those the contaminating cell, the expressions are minimized in before the transplantation in RNA level. And using this protocol, and we can freeze our cells in the dopamine cell product and to generate off-the-shelf cell product. It means once we have this off-the-shelf cell product, and we can use this cell whenever we need, because it's frozen, and we can directly transplant this frozen cell to the preclinical, the animal model. Also now to the patient. And this is the Parkinson's disease model. As you can see, we can eliminate the dopamine neurons in one side of the brain. And as you can, this one is staining with the TH, dopamine neuron marker. So you can appreciate that here is absent of the dopamines. And using this using this animal model, and we can inject amphetamine to activate dopamine and result in this rotation phenotypes. Only one side, and they rotated in only one side because this mouse has only one side of the dopamines are activated. But once we inject human stem cell based dopamine neuron to here, as you can see, you can appreciate that the dopamine neurons are rescued. Also, and also the Parkinson's disease behavior, the rotation phenotypes are rescued. And based on this protocol, we are able to generate the real clinical lot and real clinical loss of the dopamine neurons from human pluripotent stem cell. And after extensive safety, toxicity, and biodistribution efficacy test, we finally obtained FDA clearance for the clinical trial to 12 Parkinson's disease patients in the United States. And also we completed those surgical transplantations. And after 1 year post-transplantation, we announced that the phase 1 clinical trial met the primary endpoint that include the safety and efficacy with no serious side effect, and motor symptoms are improved. And if you are interested in this clinical trial, and 2 months ago, this clinical trial, the result was published in the Nature, so you can have a look at this one. And now these clinical trials are underway for phase 3 study, and if it's successful, this one will be the first case of human stem cell-based cell therapies in the world. And also, we are working on the next generation of the cell product for Parkinson's disease patient, because first, there are some needed room for the improvement. And first one is current strategy to the injection is we used about progenitor, dopamine neuron progenitor. So we cannot guarantee about the maximum the safety. The second one is more than 90% of the cells are lost during the transplantations. So we needed to improve the survival. So question is, can we further the maximizing long-term safety? And for this one, post-mitotic neuron graft is highly desired for the safety. And second question is, can we define the factors that limit initial survival of dopamine neuron graft? And once we address these two questions, final question is, can we establish the clinical relevant strategy to functionally improve the graft survival? First, to address this one, first we test whether the pure post-mitotic dopamine neuron grafting is possible. To test this one, we generate the report line, that is neuron-1-driven GFP expression cells. And neuron-1 is immature dopamine neuron marker and post-mitotic. And using this stem cell redifferentiation to the dopamine neurons, when we isolate GFP expressing cell, and followed by we performed single cell qPCR. And the resulting shows that the GFP expression cells, the GFP expression cell express the TH, the 100%, nearly 100%, and also GFP expressing cell, as you can see, appreciated that in the culture, very pure dopamine neuron population. Also, more importantly, the robust survival post-transplantation, indicating the grafting pure post-mitotic dopamine neuron is feasible. To identify the factors that limit dopamine neuron graft survival, we've performed in vivo CRISPR-Cas9 screenings with 150 genes. 150 genes contain cell death-related pathways such as apoptosis, inflammation, and autophagy, and so on. And for this one, we used inducible CRISPR-Cas9 the system, and from this one, we also include the generate guide RNA library pool, and using this pool, we differentiation to the dopamine neurons, and we isolate GFP expression, the post-mitotic cell, and we inject those cells to the mice brain. After 1 month post-transplantation, we take out, sort out, injected and survived cell. Then we perform the barcode sequencing to identify the guide RNA ratio before the after transplantations. Basically, the resulting shows that without the guide RNA, without the DOX expressions, there is no the guide RNA change. But during the differentiation in the culture, BCL-X is depleted, indicating BCL-X is important for guide RNA, the dopamine neuron inductions from stem cell. But more important one is in vivo one, and we found that the several 9 genes are enriched in the 9 genes are enriched in the grafted cell compared to before the transplantation, and those gene sets are related to apoptosis and inflammation. And using these 9 genes, we performed another CRISPR-Cas9 screening, 2 more, and we found that P53 is depleted. Depleting P53 knockout cells are more enriched in grafted cell, indicating P53 is late limiting factors for the dopamine neuron graft survival. Then we further test about P53 role in dopamine neuron graft survival. Basically, we inject P53 wild type to the left and P53 knockout to the right in the same mice, and test about the graft survival. And we found that this P53 knockout showed a more improved graft survival, and without the proliferation effect because we inject the post-mitotic cell. And also this P53 knockout cells show about dopamine neuron identity expressing the TH. But as you are well known about P53, it's a very well-known tumor suppressor, so P53 knockout strategy is not feasible, not searchable for the clinical translations. So we tried to identify some upstream factors that trigger P53 inductions. And for this one, we performed about time course dopamine neuron death, time course image analysis to identify when dopamine P53 is induced, also when cell death happens. And then this time course series was 4 hour, 1 day, 3 days, 7 day post-transplantations. And we found that the P53 is highly induced at 1 day, and then the resolved at 7 days. And this P53 induction and resolve the pattern is very similar to cell death, using, as you can see, the same cell death cleavage caspase 3 expression and tunnel assay. Then to identify some upstream factors, we performed RNA sequencing because P53 is highly induced at 1 day, and we performed RNA sequencing in the 1 day post-transplanted cell. Basically, we inject a cell and 1 day post-transplantation, we re-isolate that transplanted cell, and we performed RNA sequencing and comparing to 1 day cultured cell. And in gene ontology analysis shows that the grafted cells show enriched TNF-alpha signaling via NF-kappa B pathway, suggesting that this TNF-alpha signaling via NF-kappa B pathway could be potential upstream factors for p53 induction. And to test this one, we also performed another time course image analysis using NF-kappa B inductions, and we found that NF-kappa B is highly induced at 4 hours, preceded by more earlier in the p53 induction, so it's potential for the p53 upstream factors. And also we model this TNF-alpha and NF-kappa B p53 pathway in culture. Basically, without some normal condition, there is no induction of the NF-kappa B and p53, and then we artificially add TNF-alpha and found that p65 NF-kappa B are expressed, and those expressed cells are co-expressed with p53. Then we co-treated with the TNF-alpha antibody, the Humira. This is the drug for autoimmune disease and most selling drug in United States, and acting at inhibiting the TNF-alpha in soluble and transmembrane. And we found that the TNF-alpha co-treatment with the TNF-alpha and Humira can reduce about p53 induction, induced by the TNF-alpha. Also, the p53 downstream gene and the cell death. Then we tried to identify some strategy that is clinically applicable. And first one, we tried to identify the cell receptor, the CD markers, the surface markers, to isolate the pure dopamine neurons instead of using the Neuro1 GFP, the genetically engineered cells. And for this one, we performed the surface marker screening with using the 387 to match about Neuro1 GFP expression. So the CD marker, yes. So we found that some 2 negative marker and 3 negative marker, and actually the results that we found that at the end, 49 low and 185 high, the CD marker can purify the GFP expressing cell, and this purified cells show about some very homogeneous cell population in culture. Also, most importantly, and the robust survivor in post-transplantations. Then we co-treatment with this Humira that result in the large graft survival. And as you can see, the very right, you can see the adalimumab treated co-treatment of these cells show about large the graft size, and also the trending toward to more the motor rescuing effect. So summary here is we established the protocol to generate midbrain dopamine neurons, and from this the bench side we has many effort to move to bedside. Now we have some clinical trials of the 12 Parkinson's disease patient, and also we have some next generation of the cell product for Parkinson's disease. So I'd like to stop here. So it was most study was done during my postdoc in the Lawrence Student Lab. So I would like to acknowledge him. Also, I would like to thank you to my many support and many collaborators. Thank you for your attention.
Thanks for wonderful presentation. Okay, the last speaker is Professor Gonho Lee at Joseon University. He will present the toxin hypothesis and clinical evidence for the prevention of Alzheimer's disease.
Okay, thank you for your introduction. I'm actually the director of GAD cohort and at Joseon University. I'm honored to be introduced the endotoxin hypothesis and timeline result from double-blinded randomized clinical trial for the prevention of onset of AD. Actually, I'm focusing on the reverse translational research based on the GAD cohort, community-based cohort study. It's already mentioned about the Dr. So Yushin yesterday, and this study was supported by several government grants and the NIH and the Korea company. As you know, the Alzheimer's disease. Alzheimer's disease could be defined by accumulation of amyloid beta and tau pathology that eventually induce irreversible brain damage and cognitive impairment. Before the cognitive decline, there is asymptomatic stage for more than 10 years. I believe that it is almost impossible to recover the severe brain damage by Alzheimer's disease at the late stage. Therefore, early clinical intervention at the asymptomatic stage or preclinical stage may allow us to delay both onset of dementia and AD progression. GAD code means Gwangju Alzheimer's and related dementias code. The GAD code is basically a community-based cohort, and it was initially designed to recruit individuals at the preclinical stage and of Alzheimer's disease older than 60. In order to conduct early intervention of AD at the preclinical stage, we should find out the AD patient without significant cognitive decline. That's the reason why we are trying to screen cognitive unimpaired elderly people. Using amyloid PET scan, we screened the amyloid positive case among the volunteers and then followed their disease progression until the onset of dementia. In this respect, GAD code is clearly different from hospital-based AD patient code. Over the past 13 years, the GAD code study has been maintained by 17 government projects and 2 NIH grants, with the total funding amount is $50 million US dollars. GAD study was initiated 2013, and we are still following up the preclinical AD case for more than 10 years. Around 23,000 volunteers have been enrolled so far, and over 13,000 have undergone in the variety of tests, including full-scale neuropsychological tests, blood tests, GWAS chip analysis, 3D MRI, amyloid PET scan, and so on in the same participant. This data set is the largest one in Asia as well as Korea. As you see, our database is composed of multiple big and deep data. We collect the EEG and the ERP data from the participant. In the case of the MRI, we collect the T1, T2, T2 flare, DTI, and SWI and fMR data. And also we acquire the amyloid PET scan data, oral microbiome and gum microbiome genome, and so many multimodal data, including neuropsychological assessment and the omics data and the clinical diagnosis and the genome, whole genome sequencing data. As mentioned before, we have the conducting for more than 10 years a longitudinal study on the amyloid positive individual from the asymptomatic stage to the onset of dementia. The gray bar indicate the number of the case who have undergone just one-time medical examination. The yellow colors indicate 3 years follow-up case. The orange colors indicate the 6 years follow-up. And the red colors indicate more than 9 years. As you see, the upper panel, disease progression patterns are diverse according to the individual. Among the amyloid positive case, number 1 shows stable at the asymptomatic stage. Number 2 shows slow progression. Number 3 shows is a moderate progression, and the last one shows fast progression of AD dementia across 10 years. These findings strongly suggest amyloid accumulation is not enough to trigger the onset of AD. Accumulating evidence has demonstrated that neuroinflammation plays a crucial role in AD progression as well as neurodegeneration. However, the molecular mechanism underlying the neuroinflammation that induced AD progression is still unclear. Surprisingly, the human study has demonstrated the P. gingivalis was detected in the Alzheimer's disease brain as well as cerebrospinal fluid and saliva. P. gingivalis is the bacteria also well known to produce the endotoxin LPS that strongly induce the inflammation in diverse mammalian tissues. So we analyzed LPS level in the CSF obtained from the GAD cohort patient. As mentioned before, the LPS is endotoxin, which is originate from the gram-negative bacteria that acts as a strong inducer of neuroinflammation. As you see, the level of in CSF of the AD dementia case shows much higher than in those of the control level. Similar result obtained from the plasma sample of the same subject. These findings suggest the bacterial infection may be involved in the onset of AD dementia. In addition, the other previous studies have demonstrated that LPS was strongly detected in both the gray matter and the white matter of the AD patient. More surprisingly, the LPS are also detected in amyloid plaques in the AD brain. The recently Dr. Moon's and my colleagues' studies demonstrate age-dependent accumulation of endotoxin in the brain of the 5X FAD model without any treatment. On the basis of accumulating evidence, Dr. Brown have proposed the endotoxin hypothesis of the Alzheimer's disease. Actually, the last month I invited him to Korea and shared my findings and thoughts with him. The LPS is basically originated from gram-negative bacteria that live on the epithelial tissues, including gut and mouse and lung. The bacteria and the toxin could induce not only neuroinflammation but also amyloid and tau pathology through the blood circulation and or direct infections through the vagus nerve. During the last 2 years, the several lines of strong evidence have demonstrated human microbiota cause neurodegeneration associated with the progression of Alzheimer's disease. Next, I'd like to introduce the timeline result from the double-blinded placebo-controlled clinical trial of the effects of live biotherapeutic product in preclinical AD case. In order to inhibit the pathogenic bacteria that triggers neuroinflammation, we established the library of probiotics. These probiotics are obtained from Korean traditional fermented food, including kimchi and soybean paste, and the pieces of the healthy centenarian who live in the rural area and mainly diet on natural agricultural product and fermented food. This study is actually supported by the Professor Park Sang-chul and the Centenarian Study Group of the Jeonnam National University. In our library of probiotics, we selected 3 probiotic strains showing the strong anti-inflammatory effect and antioxidant effect. Two of them belong to the Lactobacillus genus, and the other one is the Bifidobacterium. We used the mixture of these probiotic to test protective effect on the cognitive decline and neurodegeneration in mouse model and in human model. Alzheimer's disease mouse model of 5X FAD was supplemented with the probiotics or placebo ingredient for 2 months. The TG indicate 5X FAD mouse model, TG plus PB indicate 5XFAD mouse treated with probiotics, and TG plus PS is indicate possible titer serine as a positive control. And as you see, in the both water maze and the cross maze test, the probiotics fed mice shows increased cognitive functions compared to the placebo group. We also assess the amyloid accumulation in the brain tissue of the 5XFAD mice, comparing those fed with the probiotics and those on the regular food. As you see, the diet on the probiotics has reduced the amyloid accumulation in hippocampus and the cerebral cortex. Next, we demonstrate the activation of a microglia was significantly suppressed by treatment with probiotics in 5X model, and the probiotic treated group shows decreased microglia activation in both hippocampus and cortex. We also analyzed the probiotics group has shown lower level tau phosphorylation. Taken together, the oral administration of probiotics alleviated both the tau phosphorylation and amyloid accumulation, microglia activation, and memory loss in 5XFAD mice. Next, we tried to conduct the human clinical trial with our probiotics KRP-301. The human clinical trial was randomized, double-blinded, placebo-controlled trial of the effect of the probiotics in preclinical AD. We had enrolled 100 patients divided equally between the test group and the placebo group. The test group took the probiotics in 24 weeks, and while the placebo group received the regular food. Primary outcomes are the cognitive test score, including MMSE, CDR Sum of Box, and SERAD. The secondary outcomes include RCFT, blood biomarkers, depression level, and the subjective memory complaints, and the FDG PET, and the amyloid PET scan, and another of the oral, analysis of oral and gum microbiome and the blood test. This scheme is the schematic diagram. It shows the protocol of clinical trial with probiotics. We enrolled 50 individuals in placebo and test group, respectively. There is no significant difference in demographics and the neuropsychological test score between the placebo group and the test group at the baseline. This is the result on the cognitive function. As you see, the green lines represent the test group, while the gray lines represent the placebo group. As you see, the mean change of the baseline in CDR score has a significant decrease in the test group, although the MMSE scores did not reach statistical significance. We also observed a significant improvement in the test group, particularly on SERAD subject J5 and J8, which assess the visual spatial memory and the constructional skill. Furthermore, we also assessed the level of blood biomarkers associated with Alzheimer's disease, especially GPAP and p-tau181. As you see, the both levels of the GFP and p-tau181 were significantly lowered in the blood of the probiotics group than the placebo group. Especially p-tau181 was a dramatically decreased after 24 weeks from the baseline. The neurodegeneration biomarker, the NFL, neurofilament light chain, also shows a decrease in the placebo group when compared to the probiotics group when compared to the placebo group. But there is no significant change in the level of the IL-6. It means that the systematic inflammation level. Here's the overall result, a significant protection effect on the experimental group was observed in the cognitive functions, and including SERAD and CDR sum of box. In the analysis of the plasma biomarkers, level of the GFAP and p-tau and NfL shows significantly reduced by the probiotic supplementation. Furthermore, we the uptake of probiotics reduce brain atrophy in the various regions measured by T1 3T MRI. This is the summary of the result from the brain atrophy analysis. Yeah, this is the last slide. In the mouse model, we confirmed our probiotics improved cognitive function and reduced microglia activity and amyloid accumulation and the tau phosphorylation compared to the placebo group. In our randomized clinical trial, significant cognitive improvement and was observed in the SERAD J5 and the CDR sum of box scores in the probiotics group relative to the placebo group. Additionally, we observed the promising trend in improvement of MMSE, the total SERAD score, and RCF delayed recall. The level of Alzheimer's disease-related blood biomarkers, including the GFP and the p-tau 181 and the NfL, were significantly reduced in the probiotics group compared to the placebo group. This indicates potential protective effect. For these of you interested in more detailed information about AD preventing LPB, please visit our lab and currently which is currently on display. Thank you very much for your attention.
Thank you so much. Huge, wonderful human study against Alzheimer's disease. So now we will start panel discussion. All of speakers, please. Professor Lee, sit here. Yes. All of other speakers, please come here and sit this chair. Professor.
I would like to start question first. I would like to start Professor Plundran. It was. I thank you for really great talk. I think it's very impressive. In your first part of talks, you showed that the beautiful data. Many different vaccine forms actually can induce some epigenetic imprinting at the stem cell level for monocyte differentiation. So I wonder. Exactly what kind of stimuli actually can make such epigenetic imprinting at the very first of the CLP and NMP levels?
Yeah, that's a very good question. Because the vaccines are given in the arm, locally, sub-Q, but we're seeing epigenetic changes at a site in the bone marrow. And I think the stimuli has to be some sort of cytokine. I don't know which cytokine, but maybe interferon gamma or interferon alpha or some other innate cytokine that's induced very rapidly and that acts on stem cells in the bone marrow. We're looking at that, but I don't have an answer to that question. So.
I have one more question for Andra. And also, do you have any similar phenomena actually applies to your BCG vaccine experiment? Because you didn't show the data on such kind of the epigenetic imprinting.
Yes. Other people have shown this with BCG. In mouse, BCG IV has been shown to remodel myeloid precursor cells in the bone marrow. This is a paper in Cell by Mazia Devangi in Canada. And then in humans also, I think there's some data that BCG can do some epigenetic remodeling of bone marrow stem cells. But that's from a different group. We didn't look at this. So.
Thank you.
Can I? Yeah. Actually, relative about that part, your like really beautiful inspiration talks give me like, some kind of like in tumor. Actually, bladder tumor, I think you know, inject the BCG every month, every week base, for like 4 or 6 treatment is that is a basal treatment after the surgery. So actually, like that, your talk is reminding me about the BCG treatment to the cancer. So maybe it is not just infection; it could be, treatment for the cancer. So how do you think about that part?
Yeah, no, thank you for that question. Yes, intratumoral injection of BCG has been shown for bladder cancer. And so one possibility is that BCG infects alveolar macrophages, or macrophages in the tumor environment. And as you know, BCG is a live organism. It can live inside macrophages for a long time. And so if it's live, then it has antigen. So it draws in T cells that are specific to BCG inside the tumor environment. Now those T cells are making interferon gamma, and those T cells can act on other cells in the tumor environment, like NK cells and other kinds of innate cells, maybe to have this anti-tumoral effect, regardless of antigen specificity. So that's possible. That's how it's working.
That's great. And another question, you are like a wonderful talk, is very impressive. And my question is actually, you see that from TLR5 actually effective in liver tumor is very impressive. So do you see any specific subtype of like immune cells infiltrate to that liver specifically?
Actually, you're confused because I have never checked about our TLR5 ligand in liver tumor, liver disease, metabolic disease. So
No, I understand, but like since that fibrosis is occur a lot,
yes.
so do you see any like the specific macrophage or something is infiltrated there or what?
yes, actually, our disease model is just enter into the fibrosis, not cirrhosis. So yeah, but I think many types of macrophages or neutrophils might be recruited in this disease models, but that is very well known study. So we just focusing on some hepatocyte for liver lipid metabolism pathways and fibrosis pathways in hepatic stellate cells. That is quite different because TLR5 is innate immune receptors, so everybody just focusing on some resident immune cells or recruited immune cells. But my focusing on some lipid metabolism or fibrosis regulating some different functional cells can be affected by TLR5. That is a novel idea. So
It's great. Thank you.
Okay, Professor Kim Tae-wan. Yeah. Thank you for your great talk. Yes, I have two questions. First, did you inject dopaminergic progenitor cells in substantia nigra or the ventral tegment area?
Thank you for questions. We inject our cell to the putamen area, the striatoms, where the targeting area from the substantia nigra.
So that is, did you focus only motor function, not cognitive impairment?
Yes. Actually, the current strategy for cell therapies is focusing on the movement disorder.
Okay, then my final question. I wonder how long the implanted dopaminergic progenitor cells are survivable after injection?
That's a very good question. So actually, based on the clinical trials, before using the fetal tissue, once the cells are survived within 1 month, we believe that the cells are survived until 24 years. So we found that because that's why the patients have the motor rescue functions for more than 15 years without L-DOPA treatment, drug treatments, and then once that patient dies, and 24 years later, post-transplantation, we found that still the cells, transplanted cells are survived. So yeah.
Okay, thank you.
Thank you.
I have a related question to Professor Kim. May I? I'm just wondering about your injected stem cells to produce generate dopaminergic neurons, where survival rate is a big problem. But generally, many people's wondering about the unexpected migration to the different site of your stem cells. So how can control?
Yeah, actually, that good thing about these dopamine neurons is to do not have some migrating the property. So I mean, there are some migrating neurons. There is some interneurons.
Before differentiating the target dopaminergic neuron, the stem cells can migrate.
Actually, we to this answer, actually the inject that cell is very related to, is kind of very lasting, like post-mitotic cells. Yeah, so it's a progenitor. Is I think the injected cell is between the cell between progenitor and like some immature post-mitotic cells. It's very likely about the post-mitotic dopamine neurons.
I have a question to Professor Kyung-ho Choi. The therapeutic effect of CLIP and CTC CAR T cells are quite remarkable. Now, as we all know that the T cell undergo the aging related phenotype with older, like NK-like phenotypes, for example. I wonder whether the patient age influenced the CAR T efficacy. Is there any report?
Oh, I'm sorry, I don't remember that age-related efficacy on the CAR T cells.
Yes.
Yeah. No. Sorry.
All the talks were so interesting, so I have so many questions, but I will ask Dr. Gon-ho Lee. So you talked about the inflammation, neuroinflammation by dental, I think it's not only dental, but gingival bacteria. So I was curious if there's any report about the relationship between dental hygiene and Alzheimer's disease. Also similar that any gut disease with Alzheimer's disease?
Actually, there are so many evidence is that the gum disease is possibly cause AD onset, and especially periodontitis. Actually, the P. gingivalis is the main cause of the periodontitis. So when the early case or the case of AD patient got the scaling, the tooth scaling, the progression is effectively slowed down. So many the dental or the medical doctor or even neurologist try to recommend the periodic scaling, the dental scaling. So we actually focus on the control the gum disease. Is one of the best way to protect and delay the onset of AD dementia. Thank you.
Can I ask one more question?
Yes.
Can I ask a question to Dr. Plander? So I was really interested by your BCG studies. I never known that BCG vaccine is used to so many things, because I heard that BCG vaccine is not very effective against pulmonary TB actually, but if it can, it has protective effect other viral disease. I mean, it's very interesting because I think almost all Koreans get those vaccines when we are young. So I'm curious, obviously, what makes this BCG vaccine so special, makes a protective effect against other viral disease?
You're right. BCG vaccine is not the best TB vaccine. It's the only TB vaccine that exists currently, but it's not that effective against all different forms of TB. But what is interesting is that it is having all these non-specific effects on other viral infections, other respiratory infections, allergy, and also DSS colitis. And we're testing it in models of Alzheimer's and other inflammatory diseases as well. So what makes it so special? I think for respiratory infections, for viral infections, that data we showed of BCG-specific T cells in the lung that make interferon gamma, that act in a bystander way on innate cells in the lung, epithelial cells in the lung, and stimulate interferon-stimulated genes. So it's an antiviral program. That mechanism can explain why BCG is effective against a broad range of respiratory viral infection. However, we don't understand why it is effective against DSS colitis or other kinds of inflammation. It may be something to do with bone marrow effects on bone marrow myeloid stem cells that reprograms them to make them less prone to inflammation. But we're looking at that, but we don't really have an understanding of the underlying mechanism. So.
Okay, now we will open the one more questions. Professor Song?
Yeah, I have a question to Dr. Pollander. Thank you so much. What a fascinating talk. I have two questions. Number one, based on this, responses to vaccines like yellow fever or BCG, and you could predict, for example, this reciprocal responses in response to yellow fever, right? So based on those patterns, can you predict the upstream, perhaps master regulator, which may be like common in response to vaccine or distinct to specific vaccines, right? That's number one. Number two, the BCG vaccine study, I saw the mouse studies. The sting knockout also show a little bit of, I don't know, it was discussed? No. Okay, sorry. Yes, so do you have any comments about that?
Thank you. These are great questions. So all this different epigenetic changes we see with different vaccines, but we organize them into a hierarchy. So there are IRF, the chromatin loci targeted by IRF stats seems to be highly conserved with few vaccines, AP1 loci, so you can begin to see a pattern. Now, is there a common origin for these? So we think we can trace it back to stem cells in the bone marrow, common myeloid precursor cells or granulocyte monocyte precursor cells. Now, you might say, if you look, is this happening at the single cell level of stem cells? So is there one stem cell that is destined to have more, to develop into monocytes to get more IRF imprinting, and another stem cell in the same population in the bone marrow that is destined to have more of an AP1 chromatin loci? We have some preliminary data that there is this happening at the single cell level of the bone marrow. So it is almost as if the single cell progenitor population in the bone marrow itself has different functional capabilities. Why this happens and why is there this functional distinction, this different subsets of stem cells in the bone marrow, each with a different kind of epigenetic program, we don't know, but that's what the data seems to suggest. Was there a follow-up question you asked? Something else? No. Sting, yeah, you're right. Sting does show a partial phenotype, not as clear as MIDI88, but it's like 50% effect. So we haven't followed up, but of course bacteria having DNA might trigger the Sting pathway. And it's possible you can have adjuvants that activate Sting and TLR ligands as a nasal spray to induce this kind of same sort of program. You know?
Yeah, recently, TLR ligand signal pathways can activate Sting pathways, so that is shared. So yeah, I think it might be the Stings is correlated.
I mean, the reason why I'm interested in that pathway is because as Dr. Kim showed that one of the signature of immune aging is senescence, and of course it's the pathway that cause inflammation, right? The Sting, Sting pathway. So I was wondering if, in the context of aging, it may have a major role. So in other words, if you use older mice instead of young mice for BCG vaccine, you may see the higher contribution by STING pathway than the MyD88.
Okay, so time is almost finished. So time is almost closed. So we have to finish. But before finish this session, I'd like to closing remarks of Professor Sang-chul Park. Yes.
I'm very much impressed by all the talks today and yesterday, and I was very much surprised to see that our science is so much developed. So I'm really happy, and I'm really happy that Bali, you make such a time in such a busy schedule to squeeze the time to come to Korea. So I really thank you for your visiting Korea, and I wish you should keep your attention on the immunizing, and the next time please give us more information about the immunizing, since immunizing is a really key question. And when I was in the active period, I had a very keen question about the thymus aging. Why thymus involution comes so early? So in field of immunizing, I think that we have to pay some more attention about what's the role of the thymic involution and why the thymic involution comes so early. So I think the Bali, you may make some solution about that, and other people may attention about that kind of. Since Dr. Seo, she developed a very wonderful program about ovarian aging. It's a kind of world record, I think, in the human history. If she succeeds in her work, our human life will be totally changed. So menopause period can be delayed more than 10 years. Then the population system we don't have to worry about. So I'm very happy to listen to so many good talks and wonderful discussions. Thank you very much, and have a nice trip back to.
May I say,
Yes, one more comment.
I just want to thank you, Professor Cho, Professor Kyung, and the organizing committee for inviting me to this amazingly beautiful country that you have, and beautiful people, amazing culture. I'm very humbled by your hospitality and by the incredible science and the creativity of you. So I am really deeply appreciative, and it is my honor to be with you. And amazing science, as we've heard from Professor Cho, about the impact of tissues like the ovary on aging. So I think the future is incredibly bright, and it is in your hands. And so hopefully when I come back, I will hear more amazing science from all of you. So thank you very much.
Okay, thank you to all of speakers and panels and all of attendees. So thank you so much.